the pleasure at ASCO 2023 to talk about the DESTINY-PanTumor02 interim results. This study looked at Trastuzumab deruxtecan's anti-tumor efficacy and safety across patients with advanced solid tumors that were HER2 expressing. It's a well-established antibody-drug conjugate that's quite potent. We know that this antibody-drug conjugate already has anti-tumor efficacy in HER2 expressing breast cancers, both HER2 low as well as HER2 positive breast cancers, and HER2 positive gastric cancers, as well as HER2 mutant lung cancer. However, we also know that HER2 is expressed across a variety of tumor types, and there are no approved treatment options for the other tumor types, thus there's an unmet clinical need. Further, when T-DXd was in clinical trials in early phase, there was signal of activity already with multiple tumor types, including biliary tumors, salivary tumors, and endometrial cancers. So this phase 2 trial looked at the anti-tumor efficacy of Trastuzumab deruxtecan in a patient population that was advanced, selected for HER2 expression. We selected patients that had HER2 2+ or 3+ on IHC, and we used either a central test or a local test for enrollment. And we looked at 6 different tumor types. We looked at patients with 3 gynecological cohorts: cervical cancer, endometrial cancer, ovarian cancer, 2 GI cohorts, biliary and pancreatic cancer, and also bladder cancer cohort. And then we had an all-comer solid tumor cohort that excluded these diseases, and we enrolled patients that also excluded breast, gastric, colon, and lung cancer. So we were very pleased at the analysis to see that of the 276 patients enrolled, by investigator assessment, the objective response rate was 37%. These were durable responses with a median duration of response of 11.8 months. And when we looked across tumor types, there are multiple tumor types where we see drug activity with an objective response rate that were quite high, especially in gynecological cohorts, but also quite clinically meaningful in bladder cancer and biliary cancer. The one disease we had less activity was in pancreatic cancer, but there was significant stable disease. And by central assessment of the responses, there was a 12% objective response rate in that cohort. We also had done a central IHC assessment, so confirming the expression level centrally of patients that had high levels of HER2 expression, HER2 3+. Objective response rates were very high. In the overall 3+ population, it was 61%. In the majority of the tumor cohorts, it was over 50%. It was notable that the one disease type where we didn't have as much activity in pancreatic cancer, we only had 2 patients that were HER2 3+. Also notable that our overall patient population duration of response was 11.8 months. Within patients that was 3+, it was 22.1 months. The safety profile in the study was similar to what we already know. Main take-home message is the study suggests T-DXd is active in HER2 expressing cancers across tumor types and may represent a new treatment option.